Hello, everyone. Uh, Thank you for joining us. I am calling from my hotel room, as you can see, in the lovely city of Muscat, Oman. And we are having a chance to work with our friends over here right now. So I will turn it over to Scott to say a quick hello, and then we will get started.
Hi, my name is Scott Hartley. I am the CEO of Proengin. I'm located in Fredericksburg, Virginia, so probably about half the worlds away from where Christina is, but we're welcome you guys for everyone that's in attendance. We do, just as a background on some of these, we do these about 3 or 4 times a year, and they're meant to be educational and just provide some guidance on the latest trends or things that can be done, just to provide some both education and assistance on letting people know what's available, what technology is out there, and how it can assist you. So hopefully this will be helpful, and we will have some chance later on to ask some questions. So if you, as we're speaking, you come up with those, please feel free to use the app and add those questions, and we'll gladly answer those at the end.
Thank you, Scott. So my name is Christina Baxter, and I own a company called Emergency Response Tips. And I've been working closely with Proengin for many years now. This is just an instrument that we had, and it was just fantastic and easy to use, which got me asking them all kinds of questions, which started a lot of this video series. So with that, I'm going to turn off the camera and save that bandwidth for the presentation materials. As all of these seminars go, we do record them. And so everything is recorded. You will have access to the recording after the event, and everything, like past events that we may discuss, are available to you after we do the webinars at the Proengin Academy YouTube page. And that YouTube page has historical webinars that we've been doing for the last 5 years. And we try to do one, like Scott said, every 3 to 4 months to provide a free education opportunity for all of our responders around the world that we work with. So we hope this one finds you all well, and we'll get started. So today we're really focusing on a biological threat update. And when you start thinking back, we did a threat update on biologicals last time in December. In December, I very much focused on the current threat of toxins and things of that nature, where today we're going to be talking more about the emerging threat. However, if you have a high interest in some of the basic threats that we will bring up in small amounts, then please feel free to go back and watch that last video from December, where we talk in detail about each of the different types of threats in the biological realm. So just to get started, we have bioterrorism, just as a concept, is really not a lot different from naturally occurring outbreaks. What makes it different is the intention behind it. So was there an intent to release or disseminate a material, even if that material is naturally occurring or is something that we have had as accidental leaks from labs? All of them have the same result, but the problem is the intent behind it. And so this makes it really difficult for us to start figuring out where we are. Now, if we start looking at some of the different information that we're seeing lately, and part of what I want to focus on here is the future. Is the breakout coming in biology? And why is it that the bio threat is growing at such an exponential pace? And if you haven't read the report that came out last month from NATO on their science and technology trends for 2025 to 2045, they actually have a whole section on synthetic biology in the biotechnology revolution. And they talk about how the impact is going to be made probably in the next 10 to 15 years. But reality is we're starting to see that impact now. And the early impact that we're seeing is in the fact that people are starting to work in the synthetic bio realm, but not necessarily having a lot of security and defense in place for those types of materials. So we have areas where people can buy a lot of the material needed to create harm without any type of system in place to keep track of who's buying what, where, and when. This provides us with a period of time where we have to be very careful. And we have to understand that the threat can change on a moment's notice with these types of materials. And when that threat changes, it's not something that is easily to identify as what we've had in the past. So when we start looking at this, the bioterrorism effects are going to be high morbidity, high mortality, and mainly because of the aerosolized biological agents. When we start thinking about how biological agents work, remember that in an attack context, they are generally aerosolized. And by aerosolized, we mean it is going to be a liquid or a solid, which is then dispersed in the air to make it flow like a gas, but retain its properties of a liquid or solid. The problem with that is that most detector systems out there today cannot deal with aerosols. If you have a detector and it has any type of filter on the front end, whether it be to keep sand and different types of things out, or just to dehumidify the air, all of those types of filters will remove aerosols before they ever hit the detector. That creates a problem for us as all of the newer threats that we see, whether it be in the chemical realm or the biological realm, tend to be aerosols. And that's where we've been focusing a lot of our efforts. So the impact that we have is all going to depend upon the agent used. For example, botulinum is still considered to be the most toxic material known to man, far more toxic than the things like the Novichok agents that we worry so much about. The amount disseminated, when we started to think years ago about what was happening with bioterrorism and its impacts, we very much focused on the large-scale attacks that would be led by a state actor, so nation-state to nation-state attack. What we see more discussions about and chatter in the internet realm today is about targeted attacks. So instead of having an attack that was focused on a nation state, it would be focused on a smaller region. So let's say a city or multiple cities coordinated at the same time. And that's more of what we see them talking about. The problem with that is the amount of material needed to be disseminated for those types of attacks is well within the realm of possible. For instance, anthrax spores, you can fit 10,000 lethal doses of anthrax spores in 1 milliliter of sample. That becomes really problematic. So the dispersal methods also will matter a lot. A lot of this has to do with how are they being aerosolized? Are they being aerosolized as dry powders, which means they're going to flow in one way, or are they being aerosolized as liquid solutions, which has them flowing very differently? A lot of that's going to depend upon the weather and release conditions. Where I am today, over here in Oman, it's very dry and particulates will flow extremely well. Where Scott is, over in the United States, it's likely to be much more humid. And that's going to make solid particles not move as freely because the humidity causes them to start sticking together. So it really depends upon where you are and what type of conditions are coming along. Now, the impact also is very much targeting immunity of exposed populations. So for instance, if I were having pseudomallei release in India, it may not have much of an effect because it's an endemic material there. Whereas if you have pseudomallei released in the United States, you will see a huge uptick in melioidosis cases because we don't see that material naturally in the United States, with the exception of one recent incident where they've had a couple of positive measurements in the sludge around the bottom of the Houston harbor. Now, what becomes most important in a bioterrorism aspect is how quickly can we identify the attack? Because reality is, in bioterrorism, almost always it's a retrospective study, meaning that we don't know an event has occurred unless somebody claims an event. So in many cases, a bioterrorism event can occur, and it's only the signs and symptoms of the human that provide us with the information that something happened. So I really liked what Bob Kristovich from DITRA had said back in November. And he said, we're being asked to prepare for a growing chem-bio threat in the midst of a biotechnology revolution that's changing the way that we think of what's possible. At the same time, we're preparing for this growing threat. All of these other things are converging on us with things like artificial intelligence, synthetic biology. All of these things play a huge role, and how can we be ready for all of this at once? If we start looking at the traditional detection technologies in the way that we have always worked, trying to build libraries of materials, if we don't know what the material is until after it's been released, how do we make a library of it? So we're not talking about materials that naturally occurred. What we're seeing today is the realm of possible of genetically engineered threats and the things that are coming our way. So how does this work? Part of it is looking at just how infectious diseases naturally evolve. And so when you start looking at these pathogens have to get into a cell and move between cells within the host to start with. So most of the material that we're talking about is not something that can live on its own in the real world. So for instance, bacteria, they have to grow within something. They have to have some type of nutrition. Viruses can't live without a host. So when we start looking at these types of things, the only ones that really sustain themselves in the biological realm without a host carrier are toxins. And it's the toxins that are made from their chemicals that are derived from biological origins. When you think of ricin and things of that nature, they're really a chemical at the final stage of use, but they are derived from a biological source. What happens with infectious diseases is once they get within a host, they start to modify. And then they can start going between hosts, generally within the same species. Where it starts to get really problematic is when we start to see that between species movement of materials. So modifications can be made to a lot of the different materials we deal with that are generally only seen in animals or in plants to make them susceptible to move to people or between people. And that's where we're starting to see a lot of problems coming our way. So one of the things that you have to understand to get into this phase is how is gene editing happening? Unfortunately, gene editing is actually not new. It's been around for many years. In fact, it was the first time that two women were awarded the Nobel Prize together was for the CRISPR-Cas9 process. So if you haven't seen the CRISPR process and how it works, I like to think of it as something where we're literally doing a slice and replace. So the CRISPR protein is helping us make that break. So they're coming into the DNA and they're cutting it in a very specific place. But when the DNA repairs itself, you're inserting something into that genome. That may be something as simple as removing a defective gene or removing something that is causing a disease. Those are some of the great things that have come about with all of this CRISPR technology. The opposite can also happen, where you can insert something that genetically modifies or alters the DNA in such a way that it can become a new threat. The problem that we have is that CRISPR is no longer something that just has to happen in the high-end laboratories at some of these big research universities. Today, CRISPR is making its way out into the synthetic biology realm, and you can actually buy snips of DNA to be inserted for other purposes, but that could then be used for nefarious reasons. Now, when we look at the current threat, and you guys have seen this from other talks, we talk about all the different chemical, biological, radiological, nuclear, explosive threats. But today we're just going to focus in on these biological ones. And the reality is, on the toxins, depending upon where you are, the toxins will be very different. So for instance, those of you joining us from Asia, you're more likely to see abrin, where we see a lot of ricin in the United States. Nicotine is playing a role around the world, and so aren't the things like saxitoxin, tetrodotoxin, paleotoxin. So the different types of shellfish poisons, pufferfish poisons, the poisons that we see from coral, all of these things are becoming more of a problem around the world. Botulinum has always been everywhere, and a lot of that just has to do with the way people store meats and different types of vegetables for canning. But when we start to see the change right now, is in those traditional biological warfare agents, things like anthrax, and then the genetically modified, the weaponized genomics. And when we say people should be prepared today, we're saying today, at any given time, you should be ready to respond to a toxin threat. Those are happening every day around the world. What's not happening every day around the world right now are the traditional and genetically modified threats. Unfortunately, the discussions about using them has ramped up to a pace that we've never seen before. So now when we start talking about weaponized genomics, we're looking at the fact that people will be ready to implement things of this nature in 24 to 36 months. And so now is when we must be preparing for how are we going to respond to something where we have no idea what the threat is, and we've got to then figure out a brand new material and identify it. And that's not as simple as people may think. It's one thing when you have a chemical and we can look at the structural relationships and figure out how all of those play a role. But with the genetically modified biological warfare agents, it's going to take a lot more time and a lot more energy to figure out what that threat is and how we deal with it. And frankly, it's not something that we can do in a matter of hours. It's going to be a matter of days, weeks, and maybe months before we can figure out some of these things. So when we start talking about detection, in many cases from a biological release point, it is going to be the signs and symptoms of humans that are out of the ordinary, which then trigger the fact that something has even occurred. In reality, unless something like the anthrax attacks in the United States occurs, where letters are involved and there are different types of key indicators that something is occurring intentionally, you may not know that something's happening. So if we go back to things that Shrinkio was trying to do with botulinum, or we go back to even the salmonella attacks in Oregon in the United States, just remember that those types of things were not found because of somebody saying that they had done something, but were found because of signs and symptoms, and then a good retrospective epidemiological study pulling back to the fact that these people had been near one another and shared a food source on the salmonella case. But that was something that was 100% intentional. It was intended to affect a local election in a political campaign, which is just crazy to me to even think about. But when you start looking at this, we may have people coming along that are having these types of signs and symptoms, but we may not recognize them right away as a problem. So when I start thinking about that, if I have something that's a biological threat that is cutaneous or is going skin to skin, then I may see sores and lesions on skin. So think of anthrax sores and things like that if you had a dermal exposure. When I think of something that has been ingestion, I generally see gastrointestinal distress, or I see something where people are having vomiting and diarrhea and those types of symptoms. And if it's an inhalation, I start to see symptoms that are flu-like. Well, the problem we have with that is how do we differentiate it from the common flu until we start seeing longer infection periods, different types of events coming out. So detecting early is going to be our problem. The big problem we have in biological realm is that we don't have technologies readily available that help us with this. So for instance, I have 4 here on the screen that are commonly used around the world today for biological detection. We have the adenosine triphosphate test, which is literally just telling us, is there something of biological origin? Doesn't tell us whether it's a threat, doesn't tell us anything like that, but it just says there's biological material here. So if you were to scan your phone right now, your phone should have biological material on it because you've recently held it or you've held it against your face, and so there will be some skin cells there. We have the protein tests, which are just telling us if a material contains proteins. So we don't know whether it's a threat material or not. It could be egg whites, it could be a protein powder, or it could be ricin. And so when we start to look at those, we use those 2 technologies what we call detect to decontaminate. We use it, for instance, if we have a protein test that's positive, and then we disinfect or destroy what we think is a biological threat, we want to ensure that there's no longer a positive protein test, because if we destroyed it, it'll denature the protein, and the protein test will now be negative. We then use an ATP test after we have cleaned the surface of the protein-laden materials and the disinfectant. We use an ATP test to say, did we clean the surface well? So I use the ATP test just as a test to say we wiped a surface and we wiped it well. The protein test, just for protein-laden materials, did we destroy the protein that was there? So again, those are things we can only use for decon. We can't use them to determine whether something's a threat. The next group of materials that we use are immunoassays. So a lot of you are probably familiar with the immunoassay tickets that we use that are kind of the pregnancy test for biological agents. Are we pregnant for a biological material? Now, as you can imagine, those are designed and developed for a very specific series of select agents. So most of us use up to 8 different ones at a time. And we're looking for very specific things like anthrax, tularemia. If we have ricin, some people have abrin tickets, depending upon where you are in the world. And all of those things are looking for tests that are very specific to that one threat. So it's only telling us, is one of the select agents that we have a ticket for in that space? And is it in this space in a level high enough to see on an immunoassay? And in most cases, with the exception of things like ricin, the level at which they can detect is well above the infectious dose, meaning you can't clear a scene with an immunoassay if you think there actually could have been a threat material, because the immunoassay can't measure at low enough detection limits to say that that material was there at an infectious dose or not. Which just brings us to the PCR tests, the polymerase chain reaction. Most of the tests that we use today in the field for PCR run about 70 minutes in time. There are new technologies coming along that would get us PCR to smaller and smaller timeframes, but the reality is PCR still limits us to specific threat materials because we're looking for a match. We're looking for a replication of the DNA of the material on site and see if there are certain DNA fragments in that space. So for instance, the system that I use is called Biomeme, and they have like a 27 channel system, and it's looking for about 10 different threats because they're looking for very specific DNA fragments that are indicative of a threat material. For ricin, there are 3 different DNA fragments they're looking for, and those 3 together are indicative of ricin. So we're still looking at a very much a threat agent identification, but specific to select agents. And so if we don't know the threat or if the threat has been modified, we'll miss it. One of the nice things here about PCR is as it gets faster, it does help us rule out certain things in the realm of possible. And it does, along with immunoassays, give us a little bit of identification process. But again, only on those things that we know. As we come along this line, we have increasing selectivity and sensitivity. The PCR on the threats that it is detecting can detect them at levels below that which is infectious, meaning that if we were just trying to rule out anthrax, we can do a PCR sample. And if it is negative for anthrax, we can say definitively that that sample did not have enough anthrax to cause an infectious dose in a human. So we start to get very, very sensitive on a lot of these. Now, where are we going in the future? This is something, I say future, but we've been studying this. I think the first time I started working with this specific device, it's come a long way. But this one, oh, good, it must be 15 years now. And this one, if you're looking at that, the nanopore, it's the MinION. And this one is a metagenomic genomic sequencing device. And so what it's doing is taking genomic sequencing out into the field. So when we go through and we have all of these screening technologies and we can't find anything, but we know something is going on, now we have to go to the threat of the unknown biologicals. And at this point, we can actually go through and in the field today, sequence the entire genome of a new threat. The problem is, depending upon the concentration of the threat available, that can take anywhere from 1 to 10 hours on a good day. Now, it's not doing a full genome sequencing like we would have in the laboratory systems, but enough to give us something to compare to all known threats. And for the other instance is, if it doesn't match a known threat, we can start to look and say, what else is there? The problem then becomes, you still have to go back to the laboratory to start figuring out what that threat may be and what the implications are of that threat. Now, when you talk about genomic sequencing, a lot of the times when we have a new material coming in or a new genetic modification, we can actually have a very clean sample and do a full genome sequence. But when we're out in the field, we're really in the metagenomic space. And with this one, what we're happening here is that we are going to be amplifying not only the specific threat material, but all of the other material with it, which means we're going to have an environmental background of things that are going to be amplified as well. Not necessarily a bad thing, because it tells us a lot about the environment, but a very different way of doing detection. And are we at a point where we can send this type of technology out with the average hazmat team? Not even close. In fact, there's a couple of different teams around the world that are, some like for PHAC up in Canada, if anybody's from Public Health Agency Canada, probably one of the best at this in the world you'll come across. And they have been working on this for the last 15 years to make it field ready for their teams who have PhDs in microbiology working with the system. One of the other things that this is going to take, as you can imagine, is you're not doing the computing required on such a small device, which means we're going to have access to high-speed internet every time that we start doing the sequencing and trying to compare it back to other things. And frankly, it's the sequencing and the comparison, the bioinformatics pipeline that comes with these technologies that takes all the time and is what is going to cause us to not be able to use it in the field as quickly and easily, but there's still hope for the future here. So what do we do? The problem that we're going to have is we need to be able to trigger that there's something there. We can't just go out and figure out how are we going to detect things. So if we just go out and say we're going to take a sample every half an hour and then that's going to go to a lab, that is going to become so cumbersome and so expensive that none of us will be able to sustain it. So what we're looking for are there technologies out there that can trigger or cue to tell us that there is a problem. So what we're looking for is something that can look at the background of where we are and say something out of the ordinary is happening. And that something out of the ordinary cannot be explained by temporal changes, the environment, humidity. There's different things that'll cause changes. But what we're looking for is, are there technologies that can tell us that something has happened that is very specifically biological, but is also not explained by the environment that we're working in? So that's where we started looking at a lot of the trigger and cue systems. Now, obviously, trigger and cue systems are not specific to a threat. What they are doing is telling us that there is something non-specific that is happening that is outside of the ordinary. And because it's outside of the ordinary, we need to take a sample at this point in time. So trigger and cue technologies have to be near real time or real time if we can ever get there, but near real time because it's that point in time where we need the sample. If we have something that is a trigger that tells us to take a sample, but it takes 30 minutes before it is able to take that sample, we very well may have missed that material. And now we'll have to go back and try to figure out retrospectively where it would have gone, where it would have deposited, and start doing wipe samples and things and sending to a lab, which is not going to be sustainable. So why do we start off with the particle size? The nice thing about biologicals is they have a very specific particle size distribution, which puts them into that inhalable fraction of material. So when we start looking at things that are between, I say, about 0.1 microns up to about 10 microns. So when you hear about that PM 2.5 and that PM 10 on the news and the things that are going to be inhalable fractions, that's the size material we're looking for. And so one of the things that we can rule out quickly is pollen and things of that nature, because it's a little bit too big to be of interest. So the first thing we want to do is isolate the materials that are of the size of interest that can cause inhalable fractions and cause harm in a biological realm. Then we need to be able to determine whether those materials are biological in nature and whether they are related to known and unknown living and non-living bioagents. So that's where we're heading. So when we start looking at this, we have to understand the life cycle of how the biological event will occur, especially if it's an offensive use. So if we were to have an attack or an incident, and it's a non-explosive charged incident, and it's just an aerosol release, unless somebody recognized the aerosol release as something out of the ordinary and was there to visually see it, we have no idea. So we've got to start looking for some type of alert that'll tell us that we have had an event. The next thing is the alert has to be early enough that we can provide protection both to the civilian and the responding populations to ensure that we have the right capability in place. The next thing will be to identify what that threat is. It's not enough for us just to have information to say we want to protect somebody, but we also need to at some point be able to identify what the threat is so that we can figure out how do we destroy that threat. Okay, how do we decontaminate? How do we destruct? All of those things to deal with that threat material. Okay, so when we start getting out there, the detection technologies that we're looking for are things that fall in that detect to warn range. And so in this case, we're looking for materials that can tell us it's within the particle size of respirable, it's biological in origin, and it meets certain characteristics. And so whether we're using flame spectrophotometry, we're using fluorescence, luminescent types of materials, those are the types of technologies that are going to give us a detect to warn. Then we want to come along and use that detection or other detection technologies in parallel to figure out what's the level of protection we need, how do we identify it, and then can we confirm that the way that we're decontaminating it is appropriate. For instance, if I have something that is a spore-forming material, like anthrax or even C. difficile, those things require a completely different decontamination process than something that's viral in nature because they're much hardier. We need to break through that outer shell coating to be able to get into the material to then destroy it. I'm going to turn it over to Scott and let him start going through a little bit of solution sets.
Thanks, Christina. First, I just wanted to kind of just stay on this slide and hit on one of the challenges from a detection standpoint, especially when it comes to biological and now with these genetically modified materials that potentially could come in and be threats that we need to look to address. It adds a lot of challenges, and it's really a system level approach that's going to be needed in order to detect these, and most likely involving more than one detection technology in doing that. And in some ways that it's analogous to a chemical threat from the standpoint of there is no one detector that does every single thing that you need from a chemical detection standpoint. You need to pull out the appropriate tools that'll address the threat or that can see that threat in an operational relevant manner. But also you want to get the same answer from multiple devices at the same time to give you that confirmation of what you're dealing with and the validation of it with it. In the case of chemical and biological, even though they're both threat environments, in some cases they're very divergent from one another. So in the case of a chemical threat, you're normally looking for something that's not normally there. So, you're looking for, I have some sort of nerve agent or some sort of thing or from a hazardous response standpoint, I have a ammonia leak or a hydrogen sulfide or a hydrogen cyanide leak. So it's something that's normally not there that you are able to detect. In the case of biologicals, it's much more complex because there always are biologicals present. and you are never going to change that. So the challenge becomes, how do you then differentiate between certain levels of naturally occurring or environmentally valid levels of those materials, and diverge between some sort of purposeful release. So in this case, that's what we're most concerned about, are these purposeful releases where some sort of person has incubated a decent quantity of these materials and then purposefully releases them. So there's going to be a unnatural occurrence that occurs there from an intensity level of biologicals that's not normally present, and that is where we need to be able to sample those materials and use that sample to be able to do some of the identification and validation, and then validate the decon post, because you'll have information relative to its exposure. So that's one of the challenges that you're dealing with, that makes it very unique from the chemical side of the house on there. So what we're going to talk a little bit more today is flame spectroscopy as one of those tools to help do that, and one where it's probably less known or recognized, especially on the biological side of things, on how we do that. So what I'm going to do with the next slide is just a quick video that we have that'll show you the process of how the flame spectroscopy works, and then we're going to talk a little bit more about how that then works, or how we use the tool to decipher, or for us to be specific to biological and chemical threats. So if you want to go ahead and click that, Christine, I'll walk us through that process. And I have used this on my other videos that have been present in there, but to go through there should be a click to start on there. There we go. So what you're seeing here is the inside of the detector, and there's a pre-burner, which is what you're seeing here. So because we do use a flame inside, and you can be operating in a hazardous environment, there sometimes can be material that you don't want to go outside of it, or if you're in a flammable environment. So with a pre-burner, it protects that from occurring. And what's inside the system is actually a flame. And so what that flame does is it excites the incoming airstream. What happens is those atoms become to an excited state. When they go back to their ground state, they release certain emissions specific to that element, and that element only. So what we do is have a series of lenses that captures that emissions that's coming off of the material, and then we focus it in on a CMOS sensor. So that CMOS sensor allows us to take the visible light that's produced from the thing and convert it into something that a processor can analyze and look for those fingerprints. It is both quantitative and qualitative. So it's quantitative from the standpoint of the intensity of the materials. So the amount of materials that are present directly relate to the amount of the intensity, or the amount of energy, or the amount of photons specific to that. And then each element that we're looking for has a unique emission specific to that element and that element. So it's a way of kind of describing it as a fingerprint. So all of us as humans have unique fingerprints. Same thing with some of these elements, is that they have a unique response specific to that element and that element only. So it allows us to differentiate between those. So from a chemical standpoint, which is pertinent relative, even though we're talking about biologics, in this case, in relative to your biological toxins. So those are all biologically, as Christina said, they're biologically derived materials, but they behave like chemical threats. And so in that case, they will hit on some of the different channels, and we're going to show you some videos later on in this presentation on that. Primarily, a lot of those toxin materials will hit on the nitroxyl channel, the HN channel, because they have hydrogen nitrogen bonds inside of them. But there are some materials that have disulfide bonds. So that would be your botulinum has it, and your castor beans have that, and they'll hit on your sulfur channel associated with those. So that's how we address the biological toxin materials, is that we're able to see those functional groups inside the material that allows us to identify them. So going through the same process where you're introducing the materials continuously in the airstream, applying the energy source, you're looking for a specific fingerprint, you're collecting all that spectral information, looking for that fingerprint, and then you're able to identify those elements, and then the intensity by the intensity of the emissions directly on there. So that's kind of how we handle the toxin standpoint and all your chemicals. Again, those are materials that normally won't be present. And so therefore you'll see there, and you're able to identify, hey, something's not normal here. we need to alarm and pay attention because of these threat materials that are present in there. If we go to the next slide, what we're doing in the case of your
Scott,
biological.
I just want to bring up one quick thing here.
Go ahead.
Is for us in the field, we often will use that sulfur channel to show us that we have a protein present. So that sulfur channel is similar to what we get on a protein test. So we see that sulfur bond in the protein structure. And so that has been really cool for us in field operations.
Yes, thank you. Now, if we go to the biological side, so in this case, what we're trying to do is delineate between living and non-living particles associated with that. And so what we're doing in order to add there is we're taking advantage of some additional channels that we can see through the flame spectroscopy process with that. And that's the presence of sodium, potassium, and calcium. So the reason why those are chosen is with RNA, when you have replicating materials that are present, they're using RNA to replicate them. The sodium, potassium, and calcium are key attributes that are needed for those materials to replicate themselves and that can delineate between living and non-living materials. So what we're doing is we're using the same process, collecting the information, but just on additional channels. Now, what we're doing now, or what we're introducing now, and I'm going to speak a little bit more about this over the next couple channels, is that we've recently introduced our Gen 2 system associated with this. So the Gen 1 system and Gen 2 system are still using the sodium, potassium, and calcium, but they're using that information differently. So just a little bit of background on there. So what we have done is we've done hundreds of different tests we have our own wind tunnel that we can introduce multiple materials of different types, and plus we've worked with a number of laboratories where they've released materials in there. And so we've undergone the flame spectroscopy process, and we know the specific response that we see for different materials. Now, not all materials behave similarly, so there are different ratios of these materials that are present in there, but each of those materials have a specific fingerprint associated with those. And so therefore, what we're doing is we know what those ratios are, and then we're looking for those materials in there. And that was kind of the Gen 1 system of how we went about doing that. And so if you just click on the next thing, we were able to identify an alarm based upon predetermined levels of those, looking for those specific ratios associated with those, and then we can alarm and trigger associated with that. What we're doing now, if you go to the next slide, is one of the challenges with that, especially because you're using sodium, and again, to the part of one of the earlier slides that Christine was talking about, there is some differences based upon the level or the baseline level of biological material that is present. So just taking in consideration the amount of biological material, if you were going to, in the United States, you were going to Alaska in the middle of the winter, you would have a much lower biological level of baseline in the material than you would, say, if you went to Florida or a tropical rainforest in the middle of the summer. That's just because of the ambient materials and how conducive it is for those materials to live and thrive and be present as part of background information that's present in there. The second thing is we are using sodium, and if you look to the right, you have the nice big source of sodium there that's called the ocean. So obviously, if you're close to ocean material, you're going to have a higher level of sodium in the background material with there. An additional thing relative to that as well is that a lot of our sensors are designed to operate in an environment where you are going to be changing the environment. So just think of a situation where it's a military base. You're in the middle of a military base, relatively isolated, not many people around, and your assignment for the day is to go into a city and do some monitoring. Well, the amount of baseline biological activity is going to be significantly different from when you're getting in that vehicle in the beginning of the day to where you're going to sit in the middle of the city. So all of that to say is that it's really important, and one of the things that we learned is that we have to establish a biological baseline and look for deviations from that. So those ratios that we talked about in the earlier slides still apply, but what we need to do is first establish that baseline and have a means of that baseline adjusting as the day goes on. So we're able to do that using machine learning technology. So basically what we're doing, to summarize a lot of effort that's gone over to develop the phase 2 is we're using machine learning methodologies to now take into consideration the background material and adjust to that then allows us to still use and trigger off of those biological activities to provide that upfront alarm on this. So what we're going to show here is a quick video kind of demonstrating that using the power of PowerPoint. I'm saying that a little bit like it's a good thing, but it's not necessarily the fully divergent one on that. But what we're trying to do is first we establish a sit there and establish a biological baseline, and then what we're going to do here is do a little simulation where a biological purposeful release of biological material has occurred in the middle of a city. So you're just going to see that plume go across where the sensor is doing, and you're going to see that responding on there. I think there's a little animation there if you want to try to run that, Christina. I don't know if it'll work great because you're in another country there or not, but what you're going to see there is, or what the thing would show you is that you're going to see a release, and it doesn't trigger until you see that higher level of response with it that's taking consideration of something unusual for there. But you're going to see in a normal environment, you're going to see ebbs and flows of those materials that are present, and so that's the reason why we have to have that established background, and then we trigger off of that once it reaches to a certain level above, which we normally set right now at 100 ACPLA. So that's, for those of you who aren't familiar with that, for those that you are, hopefully I'll do it justice, but from 100 ACPLA, so ACPLA stands for agent containing per liter of air. So it's a measure of biological activity in a liter of air, which would be, and so what we're trying to do is what flame spectroscopy allows us to do is delineate between living and non-living materials, and then categorize them as far as the intensity and concentration. So if your baseline is at like 10 or 20, and all of a sudden you're over 100, that's a pretty big delta. That's a 5 or 6x delta over the baseline of it. And so it's indicating that something unusual is occurring that's not necessarily a man-made or within the normal variation that you're going to see in the environment. And that's what we're going to be able to flag off of to eliminate, to basically alert and let people know that there is something unusual that's occurring in the environment. Next slide, please. So what I'm going to show you here is the AP4C-VB. So that's the product that recently got reintroduced in there with the Gen 2 algorithm. So the body of the system looks the same, but the insides have been upgraded associated with that. So this is a mill standard. This can be purchased system that's being utilized for it. So it's very ruggedized, it meets mill standard requirements on there, and it has the new algorithm associated with there. So a lot of the algorithm and machine learning is done in the processor. So the actual flame spectroscopy process isn't drastically changing from there. One additional benefit is if you are doing the flame spectroscopy process for biologics, you can then add just the additional channels for the chemical. So there is some additional processing and other things that need to get done in there, but the same form factor and body can be done with that. And the advantage therefore is you can do both simultaneously. So you can get your chemical threats plus your biological threats in one system and use that as an alarming system with it. The added benefit of this system is it is designed to be put on vehicles, for example, so you can be moving with speed. So obviously the speed and sample size is important, or the ability to handle the speed, the incoming air at different velocities is also important. This system is able to handle those as well associated with it. So it is relatively, when you start putting some of those factors in, it's a very complex problem, but the algorithms in particular are allowing us to better adjust to that environment and provide a more precise flagging environment. And we'll talk a little bit more about what that looks like in the next couple slides. So if you go to the next slide real quick. So as far as kind of your main features of your system, it's pretty easy to use. In the case of the VB, so it's both the chemical and the biological put together. We do have a product that's a bio only, so that would be called the AP4B. So it's a bio-only system, and we also have an AP4C-V, which is just the chemical thing in there. So the VB is the chemical plus the biological put into place in there. So we really have three products. They look the same, the insides are different, and the processing capability is different inside of those. with them. But they're designed, in this case, the VB is designed to handle your chemical warfare agents, your TEMS, number of TEMS and ticks, as well as your BW, biological warfare agents, to include your purposeful, your genetically modified, your traditional, and your biological toxins all put together in there with it. And it's basically, it sits there, learns the environment for a number of minutes, and then it's ready to go. Once it turns on, it's actually in ready mode within about a minute or two. And then it sits there, it learns the environment, and then it's ready to go as far as alerting and other things like that. I'll go on to the next slide and I'll explain a little bit more kind of how it gets integrated and how we now apply this or use this as part of an integrated approach. So we can click again just to hit on some of those other things on there. So design is designed to be used for can be used in ships, on vehicles. It can be used in front of command centers. It's ready to go relatively quick. So when you turn that on, the system is up and ready within 2 minutes, and it's going to sit there for about 15 minutes to search the ambient environment, and then it'll go into active presenting mode in there. So it is a flame spectroscopy tool associated with that. You can keep on clipping on there. So again, we've done a lot of tests with it. We have a system that's been outside running constantly through different seasons in different locations during pollen season, during non-pollen season, other things like that to be able to do there. An advantage of flame spectroscopy is a continuous process. There's no false alarms. And then we have the ability to integrate it to other systems. And so when it comes to biological detection in particular, that becomes very important. And so we're going to talk about why that's important by itself. So because we aren't, just in the case of the same thing with the chemical, we're looking for key materials that are present. So in the case of lewisite or arsenic, that material, whenever you're seeing it, there's nothing that really contains arsenic that's good for you. So we're alerting you, hey, there's something that shouldn't normally be here that's of a threat environment, but we aren't telling you specifically what it is, just that it contains arsenic with a high likelihood of materials with there. What we're doing in the biological in the same case is letting you know that there's unusual levels of biological activity. We aren't telling you specifically what those are as far as delineating what the specific material actually is, but then we're turning it to create a sample of that. And as Christina talked, one of the challenges that comes with biological detection in particular, that's been one of the hard things to do, is when do you take a sample? And when you take a sample and you get a hit, was it something that's just naturally occurring that I don't necessarily need to be worried about? Or is it something that is pretty important that I do absolutely need to be worried about and alarm on that? So really the advantage of the system, and these systems in particular, is they're great for sitting there continuously running and then flagging, of which you can take a sample, and then you can use your whatever your preferred method of identification is, or methods of identification for running those samples. And I'll talk a little bit more in the next slides on how that's occurred or how we're accomplishing that, which is another area where there's been some additional advances, and there'll continue to be advances going forward. So the next slide, please, on that. So what you're seeing here is what the visualization is on the system. So obviously you want to have it in a user-friendly means of being able to do there. So what we allow our systems to run as either an RS-485 or Ethernet connection. So you can do it direct, either a serial connection or an Ethernet connection. So you can either hook it up to your network or direct connection using the Ethernet port or a serial port. So we have a wide variety of means of doing that. We have displays that can hook through a serial connection that directly display the information. We have stuff that'll can be hooked on a network, so you can be monitoring it on the other side of the world if you wanted to, or you could have it within a facility where you have multiple sensors integrated all into different locations. So in many cases, it just depends on what your preference is, or you can actually use Modbus and hook it to the vehicle management system or a building management system or a ship management system on how they want to display the information on there. So in this case, we have both a biological index and a chemical index, as well as a status of where the system is, its MAC addresses, and its other pertinent information as far as cylinder levels and other things like that. And then you can capture, when you get specific captures, all of those are saved to CSV files, so comma separated files that can be brought into Excel, but we have a visualization tool as well that can be brought in there where you can see the actual data as far as concentration and intensity on those, as well as being able to understand maintenance and checking in on stuff and adjusting settings. So an additional feature that as we continue to invest in these things is we are going to, right now the systems are set for 100 ACPLA, and it takes about 60 seconds for us to trigger, where we have the potential to get down to much lower levels on that, down into the 20 level, 20 ACPLA, and much faster response time than a minute, and allowing the user to set that based upon the threat profile that they're seeing and the desired level that they want to achieve with their trigger level with it. Now, when it comes to biologics, obviously, especially depending upon what your noise environment is, you can set it potentially lower than where, obviously the lower you go, the more chance you have for triggering. So what you have to do is obviously be cognizant of your environmental variability that's present. So if your environmental variability is pretty constant and it's not varies much, you can probably get that down very low. If it's something that's going to vary very significantly, you may want to set it a little bit higher from the standpoint of to be out of those normal variations, natural occurring variations that can occur associated with the environment that you're in. The next slide, please.
One thing I just want to bring up here, Scott, is it's something that people don't really think about a lot if they're a traditional AP4C user, is that the concentration is directly being measured. We're so used to seeing the screen of the AP4C where it shows us bars and it applies that concentration into a bar, but all of the instruments are actually measuring the specific concentration. And so on the visualization tools, you can see that. And so instead of being reliant upon bars of information where you just cluster your concentrations, this allows you to see this exact concentration at any given time.
Yes, good point. Thanks for bringing that up. So as far as one of the additional areas where there's been continued development associated with there is as these system complexity is increasing, which it is only going to increase as time goes on, especially as these new tools come to the market and become more readily available, and then people with nefarious means of doing that, either purposeful or unintended consequences of those, start doing it, is the need to integrate multiple sensors and to use the sharing of data between those sensors as being able to do there. So what we have developed is a threat detection hub. So what it does is it allows us to display information on multiple detectors. Those detectors do not have to be Proengin detectors. They can be other systems that we've done. We've integrated other manufacturers of things, other detectors in there, including radiation detectors and biological or chemical, single or multiple gas chemical detectors into these things. And you can display them all in one hub in there. So you see the display there. But the heart of this is the black box that you're seeing there. So what it does is it's able to take the inputs from multiple devices, and then it basically will translate. There's some processor inside that allows us to translate that and do the output into something that our display can see and present and associated with that. So this has been used by a number of different customers with it. And our systems don't require us to have to use all of this. So we do have a known output, and we can work well with a lot of other customers. But as systems complexity is done, and some customers want turnkey solutions, this is what we have introduced out into the market in order to address that additional complexity of multiple sensors and locations, and even sensor types and sensor manufacturer, different ones, how you can display those all on a single device. Next slide, please. As a testament to this was a display that we recently did at a trade show. But what it does is it shows multiple sensors all being integrated in together. So these, in this case, we have alerts, we have a visual display, we have some single gas meters that are included. So those were not manufactured by us. We also integrated some radiation sensors. And then we have both chemical and biological sensors, as well as a sampling system. And Christine's going to talk a little bit more about the sampling systems as well. So when you're alerting, you obviously want to alert and then create a sample during that time of high biological activity. in order for you to do initial analysis on that. So all of those can be integrated together in a single system that can be monitored externally. And we've also added in cameras and GPS and weather stations and other things like that all together in an integrated approach with there. So we try to be device agnostic, so we can try to work with as many people as possible, as well as have our systems integrate into other people's systems, as well as other sensors integrate into our system, just to address the need for multiple approaches and technologies all working together seamlessly to customers to address the threat profile and the constantly changing environment that's out there. If you go to the next slide, it's just a quick animation, well, actually in 2 from now, but this is just showing how you can go back with an actual event. So this is actually showing a captured event on the system with there. So in this one had both phosphorus and sulfur on there. So you're seeing both the time and intensity of that, the real-time data that was captured onto the system. So that can be seen real time. And then you can also go back and access the actual captured data and do analysis on that as well for further investigations as far as intensity and other things relative to that, especially when if you can go back and you've done some validation on what the threat actually was, confirmation of it, in the case of a organophosphate and a chemical warfare agent, was that sarin or soman or Novichok or any of those other materials that could be present in there. And you can utilize the captured data to do further analysis as far as concentration, and that allows you to help determine with exposure levels and treatments and other things like that. If you go to the next slide, it's just a quick animation of the system and what that looks like on there. So this is a simulated, this isn't an actual event, but just a simulated on there. So what we're going to have a simultaneous release of a phosphor containing material, arsenic containing material, and a biological containing material simultaneously. And you can see what that response is. So you'll see as we run the thing, you're going to see an increase, and then it'll get above a threshold alarm. So you're seeing the real-time data coming in. Again, we're sampling and giving you the output continuous. We're doing continuous sampling and doing the output now. What you're going to start to see is we're seeing biological activity that's occurring, and we're seeing increasing levels of biological activity. So we're doing basically an integration on the incoming data that we're seeing, and as we're seeing a higher and higher intensity of that, we're giving a threat profile under the threat trend, and then we're looking for our overall bio index of that. So we're waiting for it to reach that 100 ACPLA, and we're wanting to see that over multiple seconds to get that bio alarm, and then we're triggering on that bio alarm as well as on both the phosphorus and arsenic when they exceed the threshold amounts. So in the case of the arsenic, it's at 0.5 milligrams per meter cubed, and the phosphorus is at 2 micrograms per meter cubed. So as long as those materials are above there, we're all seeing an alarm on those as shown as red on the system. And then I won't go through the full thing. There'll be a decay cycle as that goes through, and you can see the trend line go down, and then it'll release out of the threat environment of those materials. So that's how you show simultaneous versions of both the chemical and the biological materials simultaneously. And we also have information on the different sensors as far as what their levels are and some of the key parameters on there. So that just a quick demonstration of kind of what that looks like. That would occur over multiple seconds. So we did something over about 40 seconds that would probably occur over a couple of minutes. But just to give you an idea of what that looks like and how that could be utilized for an operator and someone that's monitoring things. Next slide. And just one additional thing. So our systems do run on hydrogen. So the reason why we run on hydrogen, it's one on the periodic table, so extremely light. But it also is a high and efficient burning system associated with that. So our cylinders that the AP4C-Vs run on provide about 12 hours of time of operating time on there, and the V can hold 2 cylinders, so 24 hours of runtime, and it can be hot swapped on there. We also have solutions where you can provide a higher capacity tank and just fill it into that system if you want to run for much longer periods of time. And then we also have our AP4C-F, which has its own generation of hydrogen. But for those customers that are running on cylinders, we have refill solutions that are there. Just one key point about there is their cylinders are low pressure, so it's not a high, it's not like you're dealing with a holding of a bomb in there at all. We actually have beads inside the material that adsorb the hydrogen, and they hold that into place. And so they're slowly released over time. And that's the science of kind of behind our system. Because you're dealing with hydrogen, again, that's 1 on the periodic table. You don't need very much of that. So it's really 1 gram of hydrogen over 12 hours is what it takes to run the system. So 1 gram is the equivalent of a paper clip, a little bit less than a paper clip of materials of hydrogen is all that's needed. So it's not a large amount of material that we're really utilizing with it. So it's not like you're holding a bomb inside your hand of any means at all. It's all under low pressure and slowly released as part of the system, but we have refill solutions that are available on that. And then the next slide, which I think turns it over to you, Christina.
Okay, and so to close it out, I wanted to walk through a scenario of how would this work? So in our operational context, what are we going to see? So if we were have a system in the subway, is what I had put together here, is we're going to get our background that has all the noise and the signal in there. Once that dispersal occurs, it's going to start pulling out and averaging out that background. And what's going to happen is the signal pops out of there. Because the algorithm is dynamic and it's correcting in real time for what's occurring naturally, it can easily see that signal within the noise in the background. So now we've had a signal that has told us that something is going on. Okay, so the biological alert has alerted, and now we're going to have the system take a sample for us. So there's a couple of different ones, and so I'm just looking at the integration of some of the Research International samplers. If you're familiar with these ones, these are used a lot by DoD. So we have different types of sampling systems. If you're looking at putting your sample into a liquid matrix, so let's say you want to put it into a buffer solution to use directly into your immunoassays or directly into your PCR systems, then you would use the SAS 2300. And in that case, all of the sample gets pulled in. As you trigger the sampler, it takes your sample, it collects it, it puts it into the buffer solution, and then it holds it in place. So you have 4 8 ml liquid samples ready. Those samples can be used for field analysis, they can be brought back to the lab, they can do whatever needs to be done, but you're not trying to extract a sample off of a solid matrix. Now, if you do want the solid sample, you can go with that SAS 3100 dry air sampler. These things automatically can be integrated into the AP4C products line, and what you're doing here is getting your aerosol collection. If you want them done that way, that is if you want to store the sample longer, it allows you to not have it in the liquid. So if we did have that, and I wanted to just show you if it was ricin that had, now ricin, most of the time we see it's real chunky, right? It's like what you see up in here, and that's really not much of a threat material. But if the ricin was optimized and it was put with a flowing agent like silicon dioxide or something of that nature, it was free flowing in the air for an inhalation hazard, we would be pulling it directly in through the aerosol interface instead of what I'm going to show you here, which is ricin coming in as a solid that we're transferring, because obviously we don't want to have ricin that is weaponized in the lab. So this is just chunked up castor bean here to show you where we're seeing. So you can see with the SP4F getting the high temperature as it's going, it's heating up, heating up, heating up, and then start watching your sulfur channel over here. So now we've started to see all of those bonds coming out. That's just telling me right away that, hey, this material has protein that is still viable here, and I want to further go down the testing route. So if you look over to the right, you can see where we have done a series of different immunoassays. So when we do run an immunoassay in the field, we automatically run a minimum of 2, preferably from different batches, hiding some of the codes on the top of here just for security purposes. But in this, you can see I've circled where the ricin was on each of these. And we got positive samples. We generally, with an immunoassay, have to run multiple samples, mainly because we can get what's called a matrix effect, which can block off and give us a big sample where we may not have all sample. And we have another thing called a hook effect, where we may have too much sample that pulls it right through and you don't see it. And so we always do a dilution of the sample to make sure that we're getting both things. Now, in parallel, we went ahead and ran a PCR sample, and you can see here on the right that the PCR system detected all 3 of the ricin SNPs of DNA. So they matched up to those 3. And you could also see that the system was working properly, that the internal controls made their way through the system. One is just making sure that the liquid made it through the system, the microfluidics were working. And the second one is a bacteriophage that is put in there as an internal standard to bring it through the entire process to make sure not only is the microfluidics working to bring your sample through, but the thermocycler is working to actually do your sample preparation and sample work. And so all of those things occurred. This took us about a minute to do. This took us about 15 minutes. And then we also had 70 minutes to get our sample here. Now, similarly, we actually went ahead and ran some botulinum samples here. We did not do the botulinum on the AP4C because I was not willing to aerosolize here. But what happened was we did not get detection on our immunoassay tickets. It could have been below the limit of detection, but we did get hits on botulinum on the PCR system. But we only got one of the SNPs to hit. And we did see right away that our sample prep was working properly, as well as our thermocycling system. But that allowed us to trigger to take a sample, get that sample, run the sample on the different technologies we had available, and see what we got for an answer. Now, what is that telling us? Part of what we said in the very beginning is that all of these technologies are either completely not telling us anything about the threat material on whether it's a threat or what it is when we have ATP and protein, or they're focusing in on very specific threats, but at different levels. So when I start looking at this table is just showing you the different types of technologies available and the products that are within that realm. And then if it's green, it's telling us that it's measuring at operationally relevant levels. What I mean there is if it's green, it's going to show me positive before it is an infectious dose. So you can see that we get green on a lot for a few of the toxins, but not much else. Everything else, we're having to wait till PCR to get green, where we can get actionable levels. When we get up into our handheld immunoassays, so for botulinum, we needed to have at least 500 nanograms per mil on botulinum B, or 33 nanograms per mil on botulinum A. And so those are the types of things that we're looking for to ensure that we are actually going to be able to hit those. The infectious dose, unfortunately, is well below that. Now, this is where we are. On the detect to warn is where we are just trying to get the information to tell us to take a sample. We need this to be really fast, preferably 10 seconds to 60 seconds, continuously monitoring, and allowing us to trigger a sample to be taken and then notify us that an alarm has occurred, where we can go get that sample and do something with it. Now, then we're going to take it to the identify realm, where we put it into our different technologies that are available. And so what we've been looking at here is taking something that will tell us uniquely that, hey, there's something out of the ordinary happening here. This is likely a biological living material, and it is not something that we can adjudicate from the background. And so then we're going to take a sample and go ahead and go down that molecular analysis route. And if we have to, and it's a completely new threat, then we're going to have to go down that metagenomic route with that data set, either in the field or in the lab. If we're in places like less than austere environments, if we're in a city or something, we'll take it right to the lab. If we are in an austere environment where we don't necessarily have a lab local, then we may go ahead and start trying to capture that in a metagenomic way, but allowing us to have internet access to get that data flow working is going to be critical. Scott, did you have anything to add to this slide?
No, just the fact that there are multiple technologies that are out there, and just you know, each of them has a use and a purpose, and so it's just important as part of just in the same thing with a chemical response that you understand where those technologies are. They each have limitations, and but they can be utilized as part of a holistic response.
Yes, I think we're all wishing we had that tricorder for biodetection, but right now we are so far from that it's not even funny.
Yeah.
But we do have technologies which will now allow us to say, hey, something's going wrong, gather a sample, and let's bring that for analysis. Whether you bring it to a mortar-based lab, great, if you have that capability. If not, then we may have to do some field work. Okay, with that was where I was going to end. But I did want to bring up that we had a few different questions that came in, and a couple that are right up there with things I'd like getting answers to. So one of them is, I'm trying to see how to get this to come up. Okay. One of them is there a portable version of the AP4C-VB planned?
So not presently, but it is something that we have talked about doing. The handhelds in theory have the ability to do that because we're using the same flame spectroscopy process. I think the utilization of how quickly or how fast it could change in the processing capability that it required would take a little bit of work, but in theory it is possible. It's just not something that we have gone down the full path of doing as of yet.
Yeah, it's something, Michael, that I keep terrorizing them about as well, because I would love to see a trigger system in a handheld where we could get that sample and then apply it to our other technologies, because we really don't have a good system for that today in the field. Andrew wanted to know if we were looking at replacing the AP4C with a technology like this, and then what would we do about the solid liquid samples with the scrapers? Or can this be used as a handheld unit in any way?
So the size of the V is basically, I can hold it up, it's about the shoebox size of it. So where we have, where customers have implemented or want to utilize it are choke points, for example, with a large, entrance area with a professional sporting event in front of a command and control center for the military that got set up because of an event, and they're wanting to make sure that the cloud's not coming towards them or people that are contaminated aren't coming into the control center, and the control center itself is protected. Ships, vehicles, kind of the main user point of those, and those are the applications that customers have sent to us to utilize for it. So I think it's somewhat customer focused with the solution with it, but I guess, does that fully answer the question or not? So some of it's more just,
I think to me it's more of a transportable versus portable. It's not a handheld, but it's transportable to the points of interest if there is a high priority event occurring.
And I can turn on the webcam real quick. I have it right next to me. I can actually
that would be cool.
This is it right here. So it's not very big, doesn't weigh very much associated with it, and it's here. So
Maxime's probably cringing on the other side of the camera over in France as you just picked that up and lifted it and shook it around a little bit. Sorry about that, Maxime.
Fine to shake. I'm not worried about that.
Okay. The last question we had was from Keen, and he wanted to know, how do you keep the instrument clean and working as you're probably not using a filter, which you're definitely not using?
So there is some routine maintenance that we have done with that in certain applications, that where if there was like a really dusty environment, we have a small filter that can be put on near the air intake that can then be cleaned, so it'll filter out like the large dust and other things like that. We do have a maintenance cycle that we recommend on the system. I think this one is at 18 months. Don't correct me, I should know the answer off that top of my head, but I don't. But as part of that, we do a pretty thorough cleaning of the internal components, as well as a check of the validation of it. So we expose it to each of the channels and make sure with controlled levels of gas, and do both increase and decrease cycles on it, and look over, period over the releases are over multiple minutes, and then make sure that the system is responding as it should. and we do tuning of the system as part of that. So that ensures that some of those things ensures, one, that your device is optimally running, but it also allows us to do some of the cleaning of the internal components, which can, over a long periods of time of use, can slowly degrade a little bit on the system with it.
I was gonna say, those of you who have seen some of our testing and videos, whether they're in the big lab or in the small lab, will see that we've done some pretty dodgy stuff with these detectors to see how they'll react. And thankfully, we've not yet actually harmed a sensor. So we've had to clean them, but we've not harmed one. Really cool question just came in from Tim. And he wants to know, are there systems out there that are able to detect the low amounts for mail triage in the mail systems?
So, we do have, without going into specifics as far as specific customer and locations, we do have some systems that are in mail rooms that are being utilized and are active use for that. Some of those systems are 24/7 systems. Some of them are more turn-on systems that are applied over, that are similar to the B system that you see here with them. And so the answer to that question is yes, they are being utilized, and they're in use presently in some of those applications. In some cases, some customers have certain air sampling solutions that they do to bring the air to the detector, so they can take dust off of the outside of the components or the packages and bring that into the solution as need be. So it just depends on kind of the customer and the threat level that they foresee on some of those applications.
So that was our last question. We did get a thank you from Andrew for answering his question on the handheld unit, but I have a favor to ask from Andrew. So Andrew, Scott is from South Australia, and Andrew, if you start terrorizing Scott and our friends at Proengin in France about getting a handheld unit, and I do it from our side, maybe we can make a difference and get them to make us one. So he was saying that he would love to have that as a handheld unit for their hazmat crews. And I completely agree. That would be really impressive to have something like that we could use. And so I say, just keep asking. You would be shocked at some of the things that they have done over the years just because we kept asking for it. And they're a small enough company that they're very receptive to working with the users and still providing operational realities that can change every day. So Scott, just for your information, Andrew has agreed to our plan, and so you should start receiving emails and calls from both of us. Didn't want to put you on the spot there or anything, but with that, I have no more comments or questions, but what I would like to say is we did record all of this, and we will make, as all of this trainings are, it'll be available to you at the Proengin Academy YouTube channel in the coming days. You'll also receive an information about the training as a follow-up from Proengin. And so they will be the ones contacting you. And it was nice to see, hey, Chris Moran on there. And so Chris Moran is part of the BioMeme team, and we have been terrorizing them on their PCR systems as well. And so all of these things work together, and it's fantastic seeing all of you guys working together on these capabilities to get into our hands. So thank you. I am going to close the webinar for now. And for those of you calling in from all around the world, thank you again. And we will see you on our next call, which I believe is, do you have the date handy, Scott?
I do not. Sorry about that.
I think we have one coming up in about 2 more months. So we will send that out when we send out information on this one, just to remind you that you could always sign up now for these upcoming webinars. We plan them out now, trying to do at least 6 months in advance to give you time to plan to be there. And as always, register if you can't be there, because then you'll also know right when the webinar is available on replay. So thank you. Stay safe out there, everybody, and keep up the good fight.